In this video lecture, we are going to learn about the second part of the chemistry, which is often known as like organic chemistry or biochemistry, where we are going to talk about carbohydrate, lipid, protein, and nucleic acid. These are the four major organic molecules that we are going to focus on. Okay. So let me get the PowerPoint slide for this, and then we can proceed. So as we already discussed in the first part of the chemistry, that the chemistry we divide into two groups, organic and inorganic molecules. Inorganic molecules where carbon and hydrogen is not the primary structural components. But in organic, which is what we are learning today, the carbon and hydrogens and sometimes oxygen become the part of the main compound. So organic molecule always contain carbon, hydrogen, and generally oxygen, but it can have nitrogen and other stuff as well. So carbon and hydrogen are the primary one. Okay, and we know already carbon and hydrogen, they're always linked by linked by the covalent bonds. So they are non-polar because carbon and hydrogen both attract the electron pair of the bond equally, almost equal. So they are non-polar covalent bond. Okay. And there are four main classes of organic molecule that we are going to focus on: carbohydrates, lipid, protein, and the nucleic acid. Okay. Now, this is a very interesting and important terminology. These molecules are also called macromolecules or polymer. So macro means big. So they are big molecule compared to the inorganic molecule that we just talked about, such as like oxygen, carbon dioxide, water, hydrochloric acid, sodium hydroxide. Those are like inorganic molecules. These organic molecules are called macromolecules, so they are big, or called polymer. So what is polymer means? Poly, the word means many, more means unit. So they are normally like a chain of molecules. I'll give you an example first to understand. If you look at a train, okay, whether it is like LIRR or New York subway, when you look at a train, that's a polymer. Because the train consists of multiple cars. So let's say that 10 cars are attached. So in order to learn, like if you're, let's say, learning how the train looks like for the first time, then you don't have to look at the entire train. You look at one single car and study the structure of the car. You understand all the car attached together is called train. That's polymer. So it's like a necklace. So you don't have to study all the beads. If you know one bead, that is pearl bead, and how it is it is structurally and functionally, and if all are the same, so you don't have to study all the beads. So it's like a necklace. Okay. That's polymer. In these four, carbohydrate, protein, and nucleic acid, they are polymer. Lipid is not a polymer. Lipid is like a giant molecule itself. It's like, I give an example always, like it's like a cruise ship or airplane, which is big. It probably bigger than train, but it's like a big. Okay, so that's a giant molecule, but they are not polymer, they are not chain of molecules. So how we are going to study carbohydrate, protein, and nucleic acid? This is the easy part. You don't need to study the entire molecule to know about the entire. If you know one unit particularly well, you know 99% of the entire molecule. That's our approach will be. So when we study carbohydrate, protein, and nucleic acid, we are going to find their monomer first, and we are going to study the monomer. And then we know how they join together make the polymer. So lipid is not a polymer. Now, before we proceed into any of these, we need to know something called functional groups. So functional group, as the name suggests, determine, it's like a group of molecules, a group of atoms together. They determine the main functionality of the molecule itself. Okay, those are called functional group. In organic chemistry, they are extremely important. There are many functional groups. In this course, you just need to know four of them, and I think you can do that. So it's very easy. Carboxyl group, amino group, or amine group, hydroxyl group or alcohol group, phosphate group. So carboxyl group, carbon and hydroxyl together, like think that way. Carboxyl group, COOH, bond COH. That means any other thing can attach with that COH, we call them acid or carboxylic acid. NH2 group or amine group. So if for example, in an amino acid, you have one of these at least and one of these at least. So we call that molecule amino acid. We're going to learn that later as well. Hydroxyl group or alcohol group. So when it is attached to, we call that generally alcohols or hydroxyl. Okay, we have many of them. One of them we're going to focus today in the class called glycerol. They have multiple hydroxyl group, okay, or alcohol group. And phosphate group, this is only difference about this and others are, this is particularly has a lot of charge. And this point will come to the later on. So make sure you know when there is a phosphate group attached, that molecule has a lot of charge. Okay, so PO43 minus, that's a phosphate group. So these are called functional group, as I said earlier, because these are responsible or they are the one that determine the function of the particular molecules. Once we know that, now we are going to proceed to carbohydrate. Carbohydrate is basically like the main metabolic fuel of your body. Particularly glucose is literally known as the metabolic fuel of your body. We're going to come to that glucose a little bit later, okay? So sugars and starches, you can put them together also. All the sugar molecules, that's your diet mainly. Although too much of these, we know nowadays, may cause some problem, but that's a normal US diet, okay? That's our main energy source. But they do not, like sugar, do not produce that much energy as fat can do. I'm going to mention those particular number later on, okay? But sugar is the main source. They have a 3 group we have to study: monosaccharide, disaccharide, polysaccharide. Mono means one, saccharide means simple sugar, okay? Monosaccharide means 1 unit, as I was explaining, the polymer, that train that I gave you. So we're looking at 1 car, so that's 1 car. There are 2 cars attached, and that's a train. Multiple cars attached, more than 2. Okay, so monosaccharide, disaccharide, and polysaccharide means many of them. It could be 100, it could be 1,000, it could be 5,000 sugar attached. Okay, now this does not, like sugar, although needed by your body, but they are not more than 1% of your total body weight. What does that mean? When you have excess sugar in your body transform that either complex polysaccharide format, or they convert it into the fat, okay? But sugar is required for many things, including DNA and RNA. Those are the genetic material, including them, because without sugar, DNA and RNA, your body cannot make. So let's go into a little bit more. So by the way, whenever you're studying this chapter, this is a tip. Whenever you're studying this particular chapter, let's say carbohydrate, lipid, protein, and nucleic acid, you should focus on their structure, like what they are, and their function. That's basically the chapter. So you need to know, as we just discussed right now, that these are the general function of carbohydrates. Now we're going to go into a little bit structure. So as I said, monosaccharide, disaccharide, and polysaccharide. So monosaccharide, they generally contain from 3 to 7 carbon atoms. So mostly we are going to focus today mostly on 6 carbon and a couple of 5 carbon sugar, because those are going to come back to our discussion multiple times later on as well. So this simple monosaccharide, those 3 that you must know, those are 6 carbon sugar, sometimes often call them hexose. Hexa means 6. Glucose, fructose, and galactose. Ribose and deoxyribose, they are called pentose because they are 5 carbon sugar. Okay, so glucose, fructose, and galactose. Glucose, that's basically found in, just an example, like syrup and honey. Fructose found in most fruit. Galactose found in a lot of dairy products. Ribose, the reason we are learning that because that's in the RNA, and deoxyribose, that's in the DNA, those 2 molecules. Okay, now come back here one more time. So glucose When you study in the book, you will see glucose will be represented by two types of structural presentation. One is called so-called linear, and the other one is called circular. So this is the one that normally in the human body, most in this form, like a circular form. So although the general formula of glucose is C6H12O6, so normally in carbohydrate, the carbon, hydrogen, and oxygen ratio is 1:2:1. So C6H12O6. So 6 carbon, 12 hydrogen, and 6 oxygen, 1:2:1. That's normal in carbohydrate. So glucose formula C6H12O6 you can write down in the linear format, or you can write down in a circle format, a ring format we call them. So you don't have to, for this class, you do not need to memorize this structure. What you need to know that what is glucose and how and what its function and how it works to produce polysaccharide, those. So these examples I'm going to use to describe the second part, meaning disaccharide, but I was just showing you the picture. That doesn't mean you have to memorize this. You don't need to memorize this structure. You just need to know this is the formula of glucose, C6H12O6, because it is going to come to you later on as well. Now, we learned in simple chemistry before. In organic chemistry, this is interesting, that the structure becomes extremely important. What does that mean? Like in any biology, structure is the important part. So glucose, same molecular formula, C6H12O6, as in fructose. Fructose also has C6H12O6. So why there are two different molecules? Because they are structurally different. So this is the glucose structure, and that's fructose structures. So glucose and fructose, they are known as structural isomers. Structural isomers. So glucose and fructose, they are structurally different, and their chemistry is also different. Okay, so the enzyme that can work with glucose may not necessarily work with fructose. So although both are monosaccharide, but we got to learn them separate because they found in separate area, in separate parts, in separate fruits, separate materials, and they're structurally different because their chemistry is going to be also different. Monosaccharides are glucose, one more time, glucose, fructose, galactose, ribose, and deoxyribose. Now what is disaccharide? Disaccharide when two monosaccharides join chemically. So in this example, glucose and fructose, when they come together, they lose a water molecule, H2O. They lose a water molecule and they create a bond between these two molecules. That's a bond. So when they create that bond, this reaction is called dehydration synthesis because they're losing water molecule. And when a disaccharide in a proper enzyme can cut this bond, make them two monosaccharide back again, and using a water molecule. That's why the backward direction reaction, we call them hydrolysis in this case. So dehydration synthesis, when you lose water molecule and join two monosaccharide, create a disaccharide. Hydrolysis, in the other hand, when you break down the disaccharide into two monosaccharide using the water molecules. So when you eat sucrose, for example, sucrose is nothing but table sugar. Your body will break this bond. Your body will break this bond using a water molecule, create these two monosaccharide, and then body can use this monosaccharide because the body cannot use this directly. So they break this bond, create this, and make it a regular 6-carbon sugar, and then they use it. Okay, so there are 3 disaccharide you should know. Although once they join, they are not glucose and fructose. They are totally a different component. They are totally a different compound. In this case, sucrose. So sucrose made by glucose and fructose. Maltose made by 2 glucose molecules join chemically, losing water. Lactose join 1 glucose and 1 galactose molecule chemically, and those are the 3 disaccharides: sucrose, maltose, and lactose. You must know all these 3 disaccharides. Okay. Now, this is important also. Sometimes those kind of questions can come. So follow me here. Glucose, C6H12O6. fructose, C6H12O6, when you join them together chemically, you're losing one water molecule. So what will be the formula of sucrose? Sucrose will be C12 because 2 6s make C12. H12 plus H12, H24. But look at here, H22. Why? Because we lost 2 hydrogen here. And then oxygen 6 plus 6, 12. Look at here, 11, because we lost 1 oxygen there. So what will be the sucrose formula? You add fructose and glucose minus H2O. So for every bond they create, they lose 1 water molecule. So if there are 3, just an hypothetical example, if there are 3 glucose molecules joined, so C6H12O6 plus C6H12O6 plus C6H12O6, then you have 3 carbon joined together. The formula will be 6 plus 6 plus 6, 18. So C18, then H12 plus 12 plus 12, H36 minus 4, because 2H2 is losing, because there are 2 bonds. So then it will be HC18, H32, 36 minus 4, and then oxygen will be again 18 minus 2, because 2 water molecules are lost. So whenever you get a chance, take a pencil and a paper, make sure you write down those and do the math. For every bond, 1 H2 is lost. So for 2 bonds, because 3 of them are attached, so you need 2 bonds, 2 H2 is lost. So if I add 10 glucose molecule, there will be 9 bonds. Total 10 glucose molecule, but 9 bonds required to join them. So 9 H2O will lose. So keep that in mind. The total number of molecule minus 1, that many water molecule get lost, and do the math accordingly. Polysaccharide, when you join them all, meaning join them many. So it could be 10, could be hundreds of monosaccharide, joined chemically again, losing H2O. So all the joining we call dehydration synthesis. There are 3 molecules that you must know: starch, glycogen, and cellulose. Starch and glycogen and cellulose, all 3 are chain of glucose, such as like this. This is an example of glycogen. These are glucose molecules that join dehydration synthesis. So 1, 2, 3, 4. So if just to understand one more time, 1, 2, 3, 4 glucose join. So they have 1, 2, 3 bonds. So that's what I was discussing previously. So these are glycogen molecules. So what is the difference? The difference in the pattern of their chain and pattern of their bonding. Without going in detail organic chemistry, just to make sure you understand, starch, glycogen, and glucose, all 3 are chain of glucose, but they are attached differently. They are attached differently. That is why they are 3 different molecules. So not only their pattern is different, they are even attached differently in some cases. Okay, because cellulose is attached, the chemistry is losing water, but attached a little bit differently. We are not going into that organic chemistry detail, and you don't need it. Okay. So do we generate 3 different molecules: starch, glycogen, and cellulose. Starch, why it is important? Because plants, they store their excess glucose. As I said earlier, excess glucose is not good. So they store their excess glucose in the form of starch. For potato is an example of starch that plants store. We eat them. So when we eat them, we break them down, and we have the single glucose molecule. We absorb them and use it as an energy molecule. Glycogen, we do it. I mean, we store in the form of glycogen. So when we have excess glucose, we are going to store them in the form of glycogen. So in our liver, in our muscle cells, we have a lot of glycogen. So when our glucose level in the blood drops, we break those reserve glycogen to get back the glucose level back into the blood, because we are not eating all the time. Cellulose is very important as far as human health, but cellulose, the main thing is they're making the cell wall of the plant. That's again a glucose polysaccharide, but that's required for making the cell wall for the plant. But why it is important? Because they serve as a dietary fibers. We do not have enzymes that can digest cellulose. We do not have enzymes that can digest cellulose. Otherwise, food would be plenty. Like if we could have digest cellulose, just think about it. You go outside, eat some grass and come back down, because those are cellulose, tons of cellulose, tons of sugar. Unfortunately, we can't eat them because we do not have the enzyme called cellulase, which break down cellulose. So this is all about carbohydrate that you got to know. Next is lipid. So lipid is not a polymer. One more time, lipid is not a polymer. Lipid is a big molecules, macromolecules. They have carbon and hydrogen mainly and few oxygen. Compared to carbohydrate, they have very less amount of oxygen, but they do have, but they're less amount of oxygen. They're 18 to 25% of your body weight. They're mostly hydrophobic. Some fatty acid can sparingly dissolve, but mostly hydrophobic, meaning they do not like water. So they're not miscible. They are not create a solution with the water. And they can combine with proteins for transport in the blood. So lipid normally transport. So I'm sure you heard about LDL, HDL, like bad and good so-called lipid. Those are like low density lipoprotein, high density lipoprotein. So they basically, lipid and protein make a complex to travel in the body because lipid is hydrophobic itself, but they're required. So they make a lot of things. And in order to discuss those, we need to understand, let's put 5 classes, because different book will classify lipid differently. Your book classified as generally 5 classes. Those are fatty acids, eicosanoids, glyceride, steroid, phospholipid, and glycolipid. Some book use both, but I just wrote down here just to make you less confused. But when you study phospholipid, we can also study glycolipid at the same time. So before we go into the fatty acid and eicosanoid, let's go to glyceride, because if we understand this very well, we understand this and the others quickly. So glyceride, the most common glyceride in our body is triglyceride, which is normally known as like neutral fat. So what triglyceride is, the glycerol molecule. Glycerol molecule is a molecule which has 3 hydroxyl group or 3 alcohol group. When that molecule uses a backbone and 3 fatty acids join them, we call them triglyceride. So glycerol plus fatty acid is glyceride. So I'm going to go there, but before we go to that structure, let's discuss a few different things. Just a quick information. Lipid is a very concentrated form of energy. So this neutral fat can give you 9 calories per gram compared to 4 for protein and carbohydrate. So carbohydrate, that glucose that when you studied, or the protein that we are going to study next, their calorie per gram is only 4 calories. Compared to lipid, that gives like more than double, like 9 calories of energy you get it from per gram of lipid. So our body like to store even the carbohydrate, if you have extra, like to store in the form of fat, because that's a concentrated form of energy. Okay. So when the glycerol molecule attached to the fatty acid, so now the question is, what is fatty acid? Fatty acid is nothing but a long chain organic molecules, organic acid. So I'm going to use this picture to explain fatty acid first. So ignore this, just focus on this molecule. This is a long chain molecule of carbon, 1, 2, 3, 4, and if you count them, it's 16 carbon, including the carboxyl group, COOH. Remember, in the beginning, we studied something called functional group. So when you have COOH, that's carboxyl group or carboxylic acid group. And only OH, that's hydroxyl group or alcohol group. So this is a glycerol molecule. We have 3 alcohol group attached. And this is a fatty acid molecule. It's a long chain organic acid. As I said earlier, whenever you have COOH attached at the end, we call them acid molecule, such as carboxylic acid molecule or just simply acid molecule. This is an example of a fatty acid. This particular acid called palmitic acid, which has 16 carbon, C15 plus this C, together 16 carbon. Then there could be another acid called stearic acid, oleic acid. All these are different acids. Again, you do not need to memorize this. So in the question, I will not ask you questions such as what is the formula for palmitic acid, or what is the formula for oleic acid. You don't need to know that. This is just for understanding purpose. I'm going to mention them. So the acids, the fatty acid, are two types. Okay, one is called saturated fatty acid. One is called unsaturated fatty acid. What is saturated? If you pay attention here, all the bonds are single bonds. That means carbon, when are not attached to the carboxylic group, meaning when they're not attached to the functional group, all this part is attached to the hydrogen. So each carbon has 4 valency, meaning they can form 4 bonds. If you pick up any carbon, such as this one, 1, 2, 3, 4 bonds. So they are all saturated, meaning they have all hydrogen attached properly. Then if you look at particularly this molecule, this is called unsaturated. Why it is unsaturated? Because right here there is a double bond, meaning this 2 carbon is not completely saturated with hydrogen. So we call them unsaturated. Now if there is only 1 double bond in the entire molecule, we call them monounsaturated. If there is more than 1, we call them polyunsaturated fatty acid. So whenever you buy, let's say, oil for your cooking purpose, so next time, or if you have a bottle of oil, if you look into the like olive oil or any kind of oil, on the back foot level, it will tell you like monounsaturated fatty acid content, polyunsaturated fatty acid content, etc. Now, when we're discussing this, I'm going to mention one more thing here. So whenever this is this saturated fatty acid, they are like a straight line. They appears like this, straight. So if there are 3 fatty acids joined, they're going to appear like compact, like this. But if there is a unsaturation, or there is a double bond, the molecules makes a kink, like make a bend. So they make a kink or bend. So due to that structure, they are liquid at room temperature because they need more space. We learn in chemistry that solid, less space, liquid, a little bit more space, gas, like a lot of space in between. So these kind of, wherever there is a polyunsaturated or unsaturated fatty acid, that thing will be liquid. For example, oil is liquid at room temperature because they contain unsaturated fatty acid. On the other hand, butter, they are not unsaturated fatty acid. They're saturated fatty acid. That's why they are solid at room temperature, like they stand like this. So they're all of these are like saturated completely. So they are compact or solid. Now I'm going to mention one extra thing, which is you should also know this, very important, that in 90s, due to a lot of baking industry become very popular, what they used to do, because liquid is always difficult to store. That's one of the reasons what they did. They create a hydrogenated oil. Without going into detailed chemistry, they forcefully incorporate hydrogen, and they basically, this kind of molecule, it is like this, the kink, but they bend it, like making it straight. So they keep the bond, but they convert them into straight molecules. So if you just imagine this one, you are folding back to the normal, which is not natural production, man-made production. We call those trans fat because those are trans, they contain like a trans bond. Again, without going into that detailed chemistry, those trans fat are extremely bad for health. Why? Because we do not have enzymes to get rid of them, and they can cause many type of coronary arterial disease or CADs. Okay, so they are responsible for many kind of diseases, and there's a huge study in Massachusetts General Hospital back in that time. They, if I remember correctly, like they studied approximately like more than 83,000 healthcare professionals, doctors and nurses, and all of them, and they studied interesting data. They found that if your diet has like 2% trans fat, 2% trans fat, your chance of heart attack goes up by 93%. So that is why we should not consume trans fat in our diet. Okay, coming back here. So this is a glycerol molecule, that's a fatty acid molecule. When these fatty acid molecules comes here, join, exactly same reaction, dehydration, synthesis. So we are losing one water molecules, dehydration, synthesis. We create a bond. This bond is called, right here, this bond is called ester bond, E-S-T-E-R, ester bond. So that's an ester bond, right here, they created. By the way, in the carbohydrate, those bonds, when they lose water molecule, those bonds are called glycosidic linkage. Okay, glycosidic linkage, G-L-Y-C-O-S-I-D-I-C, glycosidic linkage or glycosidic linkage. This is ester bond. They lost that water molecule. So when one molecule join, we call them monoglyceride. When two join, we call them diglyceride. When three join, we call them triglyceride. That's a neutral fat molecule called triglyceride. So why I said this is important? Because once you know how we make this triglyceride, we know phospholipid and glycolipid easily, and we already know the fatty acid. So we discuss about this saturated fatty acid, we discuss about unsaturated fatty acid, we discuss about trans fat with this particular slide. It's the oils and wax is the solid at the room temperature, so obviously they contain saturated. Their main this we discuss. So basically oil and wax, both of they have their function in the body. There's oil creates like a barrier for the bacteria, and they are like a natural moisturizer for your skin. In the integumentary system, I'm going to mention that one more time, and wax you're going to learn mostly in the immune system, the second part of the ANP, where they act as a defense mechanism in your external canal, external auditory canal. So phospholipid is extremely important. So phospholipid we are going to study. I'm going to discuss all of these, and all of these from this picture. So now pay attention in this particular picture. This is the same glycerol molecule. There is a one wedge, there is a second wedge, same glycerol molecule. If you go back 3 slides ago. So this is a fatty acid attached, that's another fatty acid attached. This one is saturated fatty acid, that's an unsaturated fatty acid. It does not matter whether saturated or unsaturated. 2 fatty acid attached means diglyceride. On that diglyceride molecule, so this is the 2 fatty acid attached to this. So instead of adding the third one, making it triglyceride, what we do, we add an organic molecule. So right here, an organic molecule which contain the phosphate group. Organic molecule which contain the phosphate group, and phosphate group is a highly charged molecule. So this part become extremely charged. But if you go back right here, this is no charge at all. So this molecule is completely non-polar because there is absolutely no additional charges here. So this is non-polar molecule. But if you come right here, this become charged. That's why this portion is known as polar head, and this portion is known as tail of the molecule. So this phospholipid molecule becomes significant molecule. Later on, I'm going to mention in the cell structure more that phospholipid, very important to understand, they have 2 parts. One part is head, one part is tail. The head part is polar, the tail is non-polar. That's why this is hydrophilic because they like water. This tail part is called hydrophobic, meaning they hate water. This is required to make our plasma membrane. So in the next chapter, when we're studying the structure of the plasma membrane, the first thing, which is the key point of that chapter, is plasma membrane is made with 2 layers of phospholipid. This is how they are arranged. So both of their head is pointing towards the opposite direction. So this could be inside of the cell, that is the outside of the cell. So the phospholipid head, which is polar, they're facing the outside of the cell because outside has a lot of water and ions. Phospholipid head, which is polar, facing the inside of the cell, which has a lot of ions and water too. So together, phospholipid bilayer makes the plasma membrane along with more proteins and other molecules. We are going to discuss that completely later on the next chapter. So this is very important for following chapter, that phospholipid is basically required to make the cell membrane of every living organism, including you and I. And obviously, what is glycolipids? Instead of this group, I don't have a picture right here, but you can study from your book, if you have a carbohydrate residue attached. Glycolipids are also required in the membrane for recognition purposes. Again, we are going to talk about that in the cell membrane chapter. Okay? So if you attach a carbohydrate residue, we call them glycolipid. If we attach a phosphate group along with some organic molecules, we call them phospholipid. If we attach a carbohydrate, we call them glycolipid. If we attach a phosphate group along with this, we call them phospholipid. Steroid, which is part of the lipid also. Steroid is basically, this is the basic structure of the steroid called cholesterol. Okay, why this is important? Because based on this structure, yes, we know that excess steroid is bad, excess cholesterol is bad for the body. We know that because they can cause a blockage of the artery. We get that. But we need some. Why? Because these molecules such as estrogen, testosterone, progesterone, cortisol, all these sex hormones, and also cholesterol, also part of the plasma membrane that we are going to mention later again, is required for the body. It's a requirement. So cholesterol is this kind of four-ring structure. They make all these hormones. So excess is bad, but we need that. Again, detail does not, you don't need it. So what I did, I put together the part that you need to know why steroid is important. So steroid basically makes the cholesterol base, all the steroids are cholesterol base in the body. They create a bile salt. Detailed function of the bile salt is not required. Just remember they are needed for the body. Vitamin D, they are needed for the body. Sex hormones are needed for the body. And all the cortisol hormone needed for the body. These are all ANP2 material that they are detailed functions. So obviously, there will be no question asking about their detailed function. Finally, icosanoid. Icosanoid is a lipid that derived from a fatty acid called arachidonic acid. Arachidonic acid forms some molecules called icosanoid. That's a fatty acid. They create a molecules called icosanoids. Those are group of molecules such as, two examples, leukotrienes and prostaglandins. Both of them are required mainly in ANP2. We are going to study them. Leukotrienes and prostaglandins, their detailed function is not required right now. Okay, they're very important molecules. So the prostaglandin act as a local hormone. We are going to learn that in ANP2. So we need to know this. So right now, what I expect you to know, I expect you to know this. That what is icosanoid? Icosanoid is a lipid derivative that derived or formed from the arachidonic acid. What are the examples of icosanoids? Leukotrienes and prostaglandins. That's it. That's all about lipid. Proteins, another very important polymer because they are chain of molecules. So they are chain of a molecule called amino acid. So protein is literally that subway car or the LIR that I mentioned earlier that has amino acid attached. So all the amino acid attached one after another makes protein molecules. Okay. What are their functions? There are tons of functions because this is one of the major building blocks of our body. So they support for like a structural protein, like as inside the cell, outside the cell, a lot of structure they help you to build. They help you to move, like all the muscles that you can act, like all the movement that you produce, that because of those protein molecules. They transport molecules. So hemoglobin is one of the major protein that transport oxygen in the blood, several others. Buffer that we study, remember carbonic acid bicarbonate buffer? Proteins are also act as a buffer. Even hemoglobin can act as a buffer. So they have a tremendous capacity to maintain buffer. This is a mistake in the slide. I think I mentioned that in the other slide that it should not write, but I believe Microsoft Word changes this. So this is the right way to write pH. And I'm not going to correct this right now, but make sure when you are learning, make sure you correct this. This is the right way of writing pH. This is not the right way of writing pH. That's like a phone number. Okay, whatever. They are the enzymes. So most enzymes are protein, not all. There are some RNA molecule can act as an enzyme. Most enzymes are protein. It's extremely important because all the chemical reactions inside the body takes place with the help of enzyme. Of course, and there are some hormones which is protein molecules. So they contain your, they do a lot of regulation because that's endocrine system. Defense, a lot of antibodies. You are very familiar with the term antibodies right now, particularly due to this situation, the COVID situation, because Antibody is the one that they fight against those molecules. Also, those are protein molecules. And then obviously there are some special protein molecules such as protein that helps into the blood clotting. Again, without going into very detail of their function, this you should know. So this detail, but extreme detail not required. Whatever I mentioned in this slide is more than enough. So now we have to learn what is protein look like how it looks like and what makes them. This is what they make them, the amino acid. But before we go right there, this is basically what this, using this particular slide, I'm going to, using those pictures, I'm going to use this, I'm going to explain these slides. Okay, so I'm going to go right here. So this, let's take a look at one particular one. This is an example of amino acid. Okay, that's carbon molecule right here, NH2 amino group, COOH carboxylic acid group or acid group. Together, this is called amino acid. That's why the name. So this is a molecule called amino acid. Okay, amino group, acid group. So what happened to the other two bond of hydrogen? They can be connected either with one hydrogen, another also could be hydrogen. Now pay attention, this is very extremely important. For every single amino acid, this portion, the bottom part right here, meaning NH2 COOH H is constant. Only thing that changes is this. For only one amino acid, that is a H, hydrogen attached. That's called glycine. That's the name of the amino acid. The other amino acid, that could be anything else. This could be anything else. This could be anything else. There are 20, this is the magic number. There are 20 amino acids that are required. There are 20 amino acids that are required to make all the proteins. There are 20 amino acids that are required to make all the proteins, not only in your body, in any living organisms. Okay, that's why this particular one we refer to as R group or side chain. So in an amino acid, this portion is fixed, no matter which amino acid you look at it. Only thing, depending on the R, which is commonly known as side chain, we have 20 different varieties. Obviously 20 different names. Your book has those 20 pictures. You do not need to know their name. You do not have to memorize glycine, cysteine, tyrosine, lysine, proline, isoleucine, leucine, valine, etc. You don't need to memorize them. Just need to know a general structure of the protein, amino acid. Now, how they make the bond? This is how they make it. Again, the same. Dehydration reaction. Dehydration reaction, meaning dehydration synthesis, meaning they're losing the H2O. So this carboxyl group, so keeping NH2 on the left side, this group comes right here. That's a side chain. That's another amino acid with a different side chain. In this pictorial representation, they are showing as a square and a triangle, meaning they want to show you like two different amino acids. Could be the same also. When they come close together, they lose H2O. They form a bond. This particular bond is called peptide bond. Peptide bond. So there is two amino acid, one peptide bond. So the third amino acid will come here, the second peptide bond. The fourth amino acid comes here, the third peptide bond, and so on. So peptide bond can attach all these amino acids. So what is protein? Protein is a polypeptide, meaning there are plenty of amino acid attached together. It could be 10 of them, it could be 100 of them. Okay, so that is governed, like which amino acid will join afterward, that is governed by your DNA. That's your nucleic acid. That's the gene. So I'm sure you heard about the word gene. So the gene is the one that has the code for a particular protein, that which amino acid will come after what. Because there are 20 of them. So using those 20 amino acids, depending on the genetic sequence, we can make any number of protein. It is exactly like we have 26 alphabets in English literature, but using those 26 alphabets, think about how many words you can make depending on which one coming after what and how many times. So for example, if PEPTIDE, it's called peptide. So P comes and then E comes, then P comes and then T again, then I again, then D again, then E again. So they create a word peptide. So using those 26 alphabets in the English literature, you can use the innumerable number of words. Same thing with the protein. Depending on which amino acid comes after what, you can create a multiple variety, like many types of protein. That's exactly what body or every single living organism does. And where that signal is, that signal coming from their DNA molecule. That's the gene. So for example, hemoglobin is a protein that is in your blood. And so body has gene that code for hemoglobin. And hemoglobin happen to have more than one polypeptide chain. So they have multiple set of genes that can produce this protein. Okay. We're going to go those details maybe very later of this course. So right now, let's go back one more time. We have 20 different amino acids. Each individual amino acid, when they have two different groups, amino group and an acid group, when they come together, the reaction called dehydration synthesis, they lose one water molecule, they form a peptide bond. And a joining of all these molecules can create a chain of reactions, and a chain of molecules or chain of amino acid, we call that protein. Now, what is very important to understand, protein, although just the joining of amino acid is not possible that easily to create in a laboratory setup. Why? Because after that joining, let's say you know what amino acid will come after what, but still after the joining, the protein will undergo 4 different stages. Most of the time 4, there are sometimes 3 to 4 different stages of modification. Those are called structural modifications of protein. That's what makes the ultimate structure of the protein. They're known as primary, secondary, tertiary, and quaternary modifications. So let me give you an example first to understand what I'm saying. So for example, just an easy example, like this is a shirt made with cotton, okay? So obviously the shirt that you are wearing probably the same, made with cotton. Now, if we pluck the cotton, that's not going to make this. That's not going to be a shirt. So what are the steps we do? We take the cotton, we make a sheet of cotton. So let's assume that's a step number 1. Then a tailor will cut that sheet of cotton in a particular way, because you cannot just wear a sheet of cotton. They will cut a particular way. That's step number 2. Then step number 3, they sew them in a particular way. So that's, let's say, step number 3. And then step number 4, they put like sometimes buttons, sometimes zipper, and the logo and everything, and then they create the shirt. That's step number 4. Same thing with protein. So when you create the sequence of amino acid, meaning based on the data in the DNA, DNA is telling us which amino acid should come after what. They're telling us because we have 20 of them. So we create a sequence that is called primary structure of the protein. That's, remember, the same, creating a sheet of cotton. So these are phenylalanine, glycine, arginine, cysteine, glutamic acid, leucine. These are name of those 20 amino acids. Again, you don't need to memorize that. So those are the sequence. That is the sequence. So when you create a sequence, that's called primary structure of the protein. Exactly like the sheet of cotton. Now remember what I said? You cut the sheet in a particular fashion. I don't know how to do this shirt. The tailor, they know it. They're expert on this. Same thing they do. They are secondary modification. What is secondary? In that peptide structure. So if you go back right here, in this peptide structure, remember this oxygen, highly electronegative? They will pull the electron towards them. So they pull the electron towards them, they become delta negative. Then carbon pull that, they lost some electron, so they will take the electron from here. Then we'll take, ultimately this hydrogen will become delta positive. Excuse me. So oxygen become delta negative, this become delta positive. Without going into detail, then this peptide backbone will have a lot of hydrogen bonding. Because delta positive and delta negative, they will attract their neighbors. So this hydrogen bonding can create 2 types of modification. One is known as simple helical structure called alpha helix. The other is known as beta pleated sheet, look like a zigzag sheet. These are all based on those hydrogen bonding happening in the peptide backbone. So based on the hydrogen bonding in the peptide backbone, they can create two structures, alpha helix and beta pleated sheet. Now comes the R group, the side chain. The side chain can also form different type of bonding. The side chain can have charged molecule as well. They can form different type of bonding. They can form ionic bond. They can form covalent bond. They can form van der Waals interaction. They can form hydrogen bond. So that can create a very interesting, like complete three-dimensional structure like this. So that cut of the cotton can now, when they fold, they become a totally different structure. It's not a sheet of cotton anymore. That's what the tertiary structure. So again, primary is the sequence of amino acid. The secondary is the alpha helix and beta pleated sheet based on the peptide backbone hydrogen bonding. Tertiary, because of the interaction of the R group of the side chain. And finally, quaternary structure, which is most protein has it. Not all, but most protein has it. Because what is quaternary? Quaternary structure is protein with multiple polypeptides. So not all protein is just one single chain. Not all short come from one single sheet of cotton. Most short are colorful, like they take it probably sheet from different type of cotton. Imagine that. So most protein in our case has a more polypeptide structure. In this example, they're giving 4 polypeptide structure, which is a common example for hemoglobin. They have 4 polypeptide structures. So interaction of their subunits. So this is one of these, this is another of these. So all 4. So interaction of the subunit is basically the quaternary structure. So in order for a protein to function properly, they have to go through those modification of structures. So nowadays, that's why the definition of protein, we call them that we just do not call them protein is polypeptide. What do we call them? Proteins are polypeptide, those who have biological activity. Because if they don't go through these 4 different structural modifications, they're not going to be active. Structure of the protein is essential for protein to be active. Protein, one of the major function is enzyme. Enzyme is a catalyst. So catalyst, what is catalyst? That is going to speed up a reaction. Because the reaction, it may happen in your body, but you need to do it right now. So how can we do it right now? We're going to use enzyme. Enzyme is the catalyst. So all the catalysts that use in the body, we call them enzymes, okay? Now enzymes, most of them are protein, but not all of them are protein because there are some RNA molecule. Nowadays we know that they can also act as an enzyme. Those are called RNazyme or ribozyme. Okay, so RNA molecules are also enzyme. That is why we cannot call all the enzymes are protein. But most enzymes are protein. That is why we're studying this right here. So what is enzyme does? 2 very important things. Enzyme does not become the part of the reaction. Enzyme, they don't become the part of the reaction. What they do, they just control the reaction. They speed up the reaction. Now, the question is how they do it. They do it by lowering the activation energy. They do it by lowering the activation energy. Now, what does that mean? Every reaction, let's look at this graph. Every reaction, they have reactants. Let's say a reaction is A plus B equal C plus D. So A plus B is the reactants and C and D are the products. So A plus B combined together, A plus B combined together makes C and D, okay? Because C and D are the products. So reactants combined together create products. Now pay attention to this one. So reactants, when they combine, they break their old bond. Whatever the bond is, they break their old bond, they make some new. And when they make some new, they become stable. In some cases, most cases, they become stable. because that brings the question like a spontaneous reaction and non-spontaneous reaction. We are not going that detailed. So those who have chemistry background, I know you know that spontaneous and non-spontaneous, we're not going to detail. I'm just going to explain simply what is enzyme and how it works. So I'm assuming this reaction is spontaneous. So the reactants combined together make C plus D, which is stable. This is, I will give an example. This is kind of like what you are doing. Even in this, like right now in the summer, instead of enjoying the summer, what you're doing, you're taking the classes, working hard. Why? So when you work hard, that takes a lot of energy. So when you work hard, you get a better job. You know that. And when you get a better job, you become more stable. Think exactly that way. Right now, you guys are here. So you guys are working hard. So this is an extra energy required for you. This extra energy required for you to understand ANP. And once you understand, you get a beautiful grade at the end. You get an A in the class. That's where you're stable, because better grade, we know better program, better job. So this energy that you are putting here is called activation energy. Now, what I am doing, I am trying my level best to make that energy that you need, that activation energy that you need, that working hard, to minimize them. So I'm helping you. I'm not making it zero. I'm not claiming, okay, in presence of me, you don't need to work hard, you will get an A. That's not going to happen. You still have to work really hard. I am minimizing that hard work to explaining the stuff to you. So this is what enzyme does. So in presence of them, the activation energy requires a little bit less. So enzymes, what they do, they lower the activation energy to help the enzyme. So in this chapter, I'm 100% sure if you sit down with your book, if you read them 5 times, you are going to definitely understand. So what is my job? My job is, after you're listening to this video, after you go through the PowerPoint, if you look at the book 1 time or twice, you're done. You understand it. That's what enzyme job. Enzyme does not, so I am not part of your product because I am not getting the grade. Same thing, enzyme is not going to become the part of the product because enzyme is helping lowering the activation energy. Clear? Now, what are the things that affect the enzyme? Of course, there are some other things that affect the enzymes. I'm going to come back to this slide 2 seconds later. So they affect the enzyme by substrate concentration. Substrate is another word for reactants. The enzyme concentration, temperature, and pH of the reaction. Again, I don't want to go very detailed of individually. Just remember, these are the 4 components that affect the activity of the enzyme. The temperature, that is why body temperature is always like 37.5 degree or 38 degree. We keep it normal, because if you have fever, everything goes messed up because the enzyme does not work properly. Same thing with the pH. We talked about that we need buffer to keep the pH same, because if the pH changes, enzyme changes. The reason is temperature and pH both change the structure of the enzyme. And we know structure is very important. And obviously enzyme concentration itself and substrate concentration both can affect the activity of the enzyme. Now, another way of explaining like how exactly enzyme works, we use the word called induced fit hypothesis. Enzyme is like, I'll give an example of induced fit. Induced fit hypothesis is basically when you put a gloves. If you put a rubber gloves, like nowadays we're using a lot. So if you put a latex gloves on your hand, your gloves is going to like surround your each finger tightly. And the same gloves, if I put, it's going to be my fit. If you put, it's going to, so we have one medium-sized glove can fit many individuals. One large size can fit many individuals. That's called induced fit. Like once you put it, it's going to snuggle fit on your body. So there is a theory that enzymes act as an induced fit hypothesis, meaning they can hold the substrate and they can break into 2 products. So this is an example of breaking, let's say, sucrose into glucose and fructose, which we learn in the carbohydrate. So that's, they're showing the example. That's called induced fit hypothesis. So that's all about enzyme and that's all about protein. The final part of this chapter is nucleic acid. Okay, so nucleic acid, which is, there are 2 of them, DNA and RNA. The first thing you need to know is their full name. Do not forget that. deoxyribonucleic acid, that's DNA, ribonucleic acid, that's RNA. So let's study the DNA first, because once you know the DNA, you will know the RNA very quickly. It is a polymer, so they also have a monomer. It is a chain, so they can have millions of them attached, but they are monomer. So what is a monomer? The monomer of DNA is called nucleotide, same as in RNA. The monomer of DNA is a nucleotide. Monomer is a nucleotide. So what is a monomer then? What you have in the monomer? You have three things in that monomer. You have three things in that monomer. One, you have something called nitrogenous bases, the compound with nitrogen in it. That's why the name nitrogenous bases. Then two, the sugar molecule, pentose sugar. Remember deoxyribose that we studied? So in case of DNA, it is deoxyribose. In case of RNA, it is ribose. So we are going to have that sugar. And third, a phosphate group. We studied that phosphate group. So how would they look like? I'm going to skip and I'm going to come back here. This is how they look like. So this is an example of nitrogenous base. Nitrogenous base are 4 types in case of DNA. They are called adenine, guanine, cytosine, and thymine. This example is thymine. A, adenine, G for guanine, C for cytosine, and T for thymine. These are the 4 nitrogenous bases. This is how they look like. So this is thymine look like, this is adenine look like, this is guanine look like, this is cytosine look like. So obviously you don't have to memorize their structure. You just need to know there are 4 variety of nitrogenous bases. They call adenine, guanine, cytosine, thymine. Sometimes in short we call the A, C, G, T. So these are the 4. Then there is a 5th variety, which is called uracil, very similar as thymine, uracil, which referred to as U. Uracil is only present in RNA. So RNA will have AUGC, not T. So thymine ATGC will be in DNA. RNA will have AUGC. Same thing. Okay. Now, when you're talking about adenine, guanine, cytosine, and thymine, I would also like to mention they are divided into 2 groups. One is called pyrimidines, and the other is called purines. Adenine and guanine, these two, where there are 2-ring structure, those are called purine bases. And uracil, thymine, and cytosine, uracil, which is in the RNA, thymine is in the DNA, and cytosine in both. So uracil, thymine, and cytosine, they are 1 carbon ring structure, carbon nitrogen ring structure, 1-ring structure. They are called pyrimidine. So going back here, this is pyrimidine, that's pyrimidine. This is purine, that's purine base. And uracil also is a pyrimidine. So that's all about nitrogenous base. Now coming back here. So this is a nitrogenous base. It could be either A, could be either T, it could be either G, it could be either C in case of DNA. Attached to a sugar molecule. What kind of sugar? Deoxyribose. Because in a normal ribose, when you look into detailed structure, there is another oxygen here. But this oxygen is missing. This carbon is attached with the hydrogen and another hydrogen. Okay, instead of OH. This makes it a deoxyribose. Deoxyribose, 1 oxygen less than ribose. And then it is attached to a phosphate group. This entire structure, we call it nucleotide. So this is the one, nucleotide. Now what is a DNA? DNA is a chain of nucleotide. DNA is a chain of nucleotide. It could be depending on the base, we refer to them A, T, G, and C. Okay, because this part is a fixed. Now question, how they create that bond? This is how they create the bond. Now pay attention. This is thymine in our example. That's the sugar residue or deoxyribose. This is where the phosphate group is. What they're going to be attached to the neighbor, which is in this example a G residue, like a G nitrogenous, guanine nitrogenous base, sugar, and a phosphate. So this is where they're going to attach together. That phosphate group is going to be attached to the carbon of the sugar molecule. That is why it is known as sugar phosphate backbone. So if we go right here, you can see that same thing right here. You see that nitrogenous base, sugar molecule, and a phosphate. This and this carbon is going to get attached. This and this carbon is going to get attached. So they create a sugar phosphate backbone. So when you go backwards, like when you go here, this is called sugar phosphate backbone. So this bond between the phosphate and the carbon of the sugar molecules, we call phosphodiester bond. Phosphodiester bond. So they create a sugar phosphate backbone. And that's how the chain is. Because sugar phosphate backbone, everything is same, we refer DNA as like T, G, A, C, C, A, T, T. You can write down any number of bases, millions of bases in a long DNA molecule. Okay, so now when DNA was discovered first, and then people studying their structure, they found a very interesting two things. One is DNA molecule is not single-stranded. DNA molecule is a double-stranded molecule, like this, double-stranded molecule, okay? Like it is like two sides of a ladder. Like the ladder has all the rungs here and then the two poles. That's what the DNA double-strand is. So DNA is a double-stranded molecule, and not only double-stranded, these strands are running in an opposite direction, meaning one strand this direction, the other strand is this direction. So they're running in opposite direction. Because as I said earlier, organic chemistry is all about structure, all about their spatial arrangement, all about their molecular direction. So just without going into very detail, quickly, this molecule, when you're reading this particular molecule, this is how we read that this particular carbon is number 1. That's our convention, right side of that oxygen, number 1. So one carbon, first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. That's what this is numbered. First carbon, second carbon, third carbon, fourth, and that fifth carbon. So what DNA has, the one end they will have with the 3 carbon, or we call them 3 prime, and the other side will end up with the 5 prime. So DNA goes from 3 prime to 5 prime this direction, and look at here. 3 prime to 5 prime, the opposite direction. So imagine this is 3 prime. So 5 prime to 3 prime in this direction, and then it will be opposite direction. This will be the 3 prime and then 5 prime. So they're running in, we call them anti-parallel. So 3 prime one direction, one side, 5 prime the other side, 3 prime in this side, 5 prime in the other side. So they're running anti-parallel. So that's one thing. So DNA double-stranded and anti-parallel. Second thing is, the complementary strands will be always like, if this is T, this is complemented by A. If this is G, that's complemented by C, and vice versa. So A always complement with T and vice versa. And G always complement with C and vice versa. So if you really know one strand, you automatically know the second strand. So if you know one strand is A, G, G, T, C, A, G, G, T, C, the opposite will be T, C, C, A, T, because, you know, A, G, the final one, whatever. I mean, what you should do at home, you write down a sequence, any arbitrary sequence, and try to do the complementary sequence. Complementary sequence can be easily done. As I said, A and T will be vice versa, like they will always go that way, and G and C and vice versa. Now there is a third point. A and T, between them, there's a 2 hydrogen bond. G and C, between them, there are 3 hydrogen bonds. Do not forget that. So A and T, between them, 2 hydrogen bonds. G and C, between them, 3 hydrogen bonds. So that's DNA molecule. Once you know the DNA, you automatically know the RNA, because RNA has 3 different things. Number 1, RNA is a single-stranded molecule, because they don't have to be that stable, because this is your genetic material. This is you. So that has to be very stable. RNA do not need to be that stable, because DNA temporarily create RNA, which can make the protein. We're going to learn that later on. Okay, so RNA is single-stranded. Number 2, RNA, the sugar molecule they have with oxygen, that means they're regular ribose. They have no oxygen, they're deoxyribose. This is regular ribose. And the third is, they don't have T, they have U. Instead of thymine, they have uracil. So DNA double-stranded, RNA single-stranded. DNA has bases called ATGC. RNA has bases called AUGC. DNA has DNA has a deoxyribose. RNA has a regular ribose. These are the 3 differences. Those are the 2 nucleic acids. you can have this, and then there are three different type of common. There are many actually. So without going into very detail, just remember right now, for your right now purpose is good enough. There are messenger RNA, normally known as mRNA, ribosomal RNA, rRNA, and transfer RNA or tRNA. Detail not required right now. We are going to learn them later on. Finally, this is a molecule that you must know. This is a molecule called the ATP. Okay, adenosine triphosphate. This is nothing to do with nucleic acid, protein, or carbohydrate or lipid. This is a molecule that is so-called energy currency of our body. That's an energy. So any energy related work, we say, hey, ATP. Like for example, our normal currency is like $1. Any trade we do, anything we buy, anything we sell, we use the dollar as a currency. Same thing here. Any body energy transition, there are a couple of other, but ATP is the main one. That's a main high energy compound of our body. Now, what is ATP? Like how they look like? Because we're discussing nucleic acid, we should know it's look like very similar. That's the nitrogenous base that we just decided, we just looked into it, adenine, that are attached to the regular ribose. And then they're attached to the 3-phosphate group. That's why it's called triphosphate. So adenine, ribose, regular ribose. So they are more closely related to the RNA, not the DNA, because DNA has deoxyribose. RNA has a regular ribose. This is a regular ribose. That's oxygen there. So ATP is most closely related to RNA molecule, not DNA molecule. And what is ATP again? Adenosine triphosphate. So adenine, ribose, and then 3-phosphate group attached. Together we call ATP. Now, why this is so important? The important is, as soon as you break this bond right here, as soon as you break this bond, energy can be released. And that energy can be coupled with many other reactions. So let's say you want to do something, let's say you want to do muscular activity and you need energy. So you're going to break this bond, that energy can be used to use muscle contraction. That energy can be used to for me to speak. That energy for you to use writing. So what body does, body create ATP. It's like a battery. So they're creating the ATP. How they're creating? There is an enzyme called ATP synthase. So synthesis of ATP is basically ATP synthase. What they do, they add this phosphate to the end. So if you break down the last molecule, we call them ADP, adenosine diphosphate. If you break down two of them, we call them adenosine monophosphate, AMP. So this is the whole, when you have all three, adenosine triphosphate. You break the one, adenosine diphosphate. You break the second one, adenosine monophosphate. So every time you break, you liberate energy. And that energy can be coupled. We learned that. Okay? So how do you make a new one? So when you have ADP, adenosine diphosphate, you add a phosphate at the end, you make it adenosine triphosphate. How do you add that phosphate? This is the enzyme, ATP synthase, they do it. So that is how we create our energy molecule in the body. Okay. So that is the entire chapter. There is one more slide, which basically I found in some Pearson website, so from another book. So they kind of like put everything together. And so this will be a nice review point. So in this particular chapter, when you study, make sure you know the functional group. First, only 4 functional groups that I mentioned, make sure you know all of them. Make sure you know carbohydrate, what is their function, and then monosaccharide, disaccharide, polysaccharide, what are the differences, how they make the bond, and what are the monosaccharide, what are the disaccharide, what are the polysaccharide we studied. Remember, we studied 3 polysaccharide, we studied 3 disaccharide, sucrose, lactose, and maltose, and we studied 5 monosaccharides. So make sure you know that. And this will summarize their function as well. Then we study different type of lipid. We study triglyceride, fatty acid, wax, phospholipid, oil, steroid, eicosanoid. So make sure you know all of them. Then we study the protein, the structure of the amino acid, how they join the amino acid. Then the 4 different modification, which is primary, secondary, tertiary, and quaternary. Primary is a sequence of amino acid. Secondary, the alpha helix and beta pleated sheet because of the hydrogen bond of the peptide backbone. Tertiary, the interaction of the R group or the side chain, various type of bonding, and quaternary interaction of the subunit. We learned that. Then we also learn about enzyme. Make sure you know all enzymes are not protein because there are some ribosome as well. And enzyme, how it works and what factors affect the enzyme activity. Then we study the nucleic acid, first study the DNA and then study the RNA. So DNA is the double-stranded. A is always bond with T, they have a double bond. G always bond with C, they have triple hydrogen bonds. And then the DNA has deoxyribose, they have the ATGC, these are the 4 nitrogenous base. RNA with differences I mentioned earlier, make sure you know those things. Then you should also know ATP, adenosine triphosphate, okay, which is the energy molecule. Cyclic AMP is like a little bit different than AMP. Again, you don't need that right now because cyclic AMP, we are going to use it in the, we are actually not going to use it in this. We're going to use it in AMP2. They are intracellular messenger. They uses a second messenger pathway. So right now, you really do not need to know cyclic AMP. Okay, so that's basically the entire chapter. So this is the completion of the organic chemistry, and the other video lecture we had completion of the inorganic chemistry together. You have the chemistry chapter. Okay, thank you.